Hello, everyone. This is Lindsay with Family Office and Networks. Thank you so much for joining us today. We are here and we are live. We're just giving everyone a quick last-minute opportunity to get logged in before we jump into today's presentation. With that said, we're going to get started in about 1 to 2 minutes, and we thank you for your patience. Hello, my name is Lindsay Shuck. Thank you for joining today's Family Office Network's webinar series featuring Asalyxa Bio. Before we jump into the presentation, I would like to briefly introduce our speakers. Mark Appel, CEO. He is an experienced investor in healthcare and biotech spaces. He helped to build the structured healthcare and royalty investing strategy at Marathon Asset Management, where he was instrumental on a range of large biotech deals. Prior to that, he invested across the capital structure and public market healthcare opportunities. Mark has previously held roles at Highbridge Principal Strategies, Eaton Vance, and Kirkland & Ellis. Mark received his BA from Yale University, his JD from Harvard Law School, and his MBA from Harvard Business School. He is an active member of the New York Bar. Bruce Auerbach, head of preclinical development, is also joining us today. Bruce has over 20 years experience in pharmaceutical drug discovery and early clinical development. As co-founder and president of AlphaCore Pharma, he utilized SBIRs following a successful phase 1 clinical trial. Prior to starting at AlphaCore, he was an associate research fellow at Pfizer, specializing in drug discovery and early clinical development. Bruce graduated with an MS in pathology and comparative medicine from the Bowman Gray School of Medicine of Wake Forest University. At the end of today's presentation, we will have time for Q&A. Please use your questions chat option on your screen to send over any questions you have so we can answer them accordingly. And now with that said, I'm going to hand it over to Mark and Bruce.
Great. Well, thank you, Lindsay. As Lindsay mentioned, I'm Mark Appel, the CEO of Asalyxa Bio, and Asalyxa is a platform company focused on targeting immune cells for the treatment of a wide range of inflammatory and autoimmune diseases. Before we dive into the presentation, just want to quickly highlight that we've made a lot of progress in a relatively short period of time. Over the past number of months, we've liaised with the FDA and gotten clarity on the path through and into clinical trials. We've also kicked off the manufacturing process. We should be finishing our IND enabling studies by the middle of next year and kicking off our phase 1 study also in the middle of next year, and should have phase 1 data readouts by later next year, and should be in phase 2 by early 2022. So a lot happening in a relatively short period of time. Next slide. This is just our forward-looking statement. Next slide. So before we dive in, let me give you a brief background and a little intro through an investment overview. First off, we're going to be talking a lot about our Panther platform, and that's the platform technology that allows us to target specific immune cells, particularly neutrophils. We're going to talk also a lot about the lead program on the Panther platform, which is ASX100. And ASX100 combines a very strong safety profile from the fact that these are very well established therapeutic components, combined with a very broad range of therapeutic opportunities that we can pursue. And that program is advancing very rapidly towards clinical development. We'll also talk more about the range and versatility of the Panther platform that should allow for rapid identification and advancement of multiple other therapeutic programs to target a range of immune and inflammatory diseases. Both Panther and ASX100 have a robust patent portfolio behind them that extends to nearly 2040. And with the Panther platform, there are a range of high-value exit pathways here, both through licensing of specific programs, licensing the platform for use for other pharmaceutical companies to use on their programs, and also IPOing the company. Next slide. Here's the Asalyxa leadership team. On the business side, I'm not going to reintroduce myself, but I'm additionally supported by several key members of my team. Greg Licolai, who is the Chief Medical Officer of PRA, which is a very large clinical research organization. He's also a former executive of Moderna, which obviously is a company that's gotten a lot of attention in recent months. I'm also joined by our COO, Ryan Fox, who spent many years at GSK overseeing operations and R&D there, and by Ken Olivier, who spent many years in toxicology, both at large pharma and at some pretty high-profile biotech companies like Morphic and Torque, and has led multiple programs through IND and even some through to NDA. On the scientific side, our scientific leadership is headed by Dr. Lola Eniola Adefeso, who's also the academic collaborator and founder of the technology. She's a professor of chemical engineering and biomedical engineering at the University of Michigan, and she's devoted most of her career to research around targeting of particles to different immune cells with a real focus on neutrophils, which really ties in very closely with the activities of the company. Additionally, we're supported by two serial entrepreneurs who are key members of our team, John Freshley, who's the head of corporate development and was previously the CEO of multiple companies in Ann Arbor, where Asalyxa is based, including ONL Therapeutics, and Bruce Auerbach, who you have just introduced previously and will be speaking about ASX100 today. Next slide. Now I'm going to briefly introduce the Panther platform, and this is really what the underlying technology that is the basis of Asalyxa. And what Panther are microparticles that are made up of a polymer, which is made up of two main ingredients, salicylic acid and adipic acid. And salicylic acid is the active ingredient in aspirin, and adipic acid is a food additive that's known to be safe by the FDA. And so what happens is we take this polymer and we are able to wrap it up and divide it into different shaped and sized microparticles. And that's really the basis of the Panther technology, because that range of size and shapes allows us to target different immune cells and get drug into different immune cells to target specific immune responses. Additionally, we can actually lace other drugs into these particles and that way load them with other therapeutics. So although the active ingredient in Panther unloaded is salicylic acid, we can actually put more potent therapeutics into these particles. And we'll talk to you a bunch later about ASX100, which is the first program in that platform. Next slide. In terms of safety, I just want to emphasize what gives us confidence that the Panther platform is so safe. First, the underlying components. Salicylic acid is an active metabolite of aspirin. It's been in people nearly over a century at this point. Many clinical studies done with aspirin before, all generally known to be safe, particularly at the doses that we're talking about. Additionally, adipic acid also tested extensively in people. It's a food additive, very little risk for concern there. And those are really what this ultimately breaks down into. The polymer that's made up of salicylic acid and adipic acid, poly A, has been used as a coating in stents, so it has been in people before as well. So that gives us confidence that the polymer is safe. Although the particles themselves, the Panther particles, have not been tested in people yet, that's something that we're intending to do in our clinical trials. Particles of much greater size have been used safely in people before, and the poly A particles have been tested in human blood and show no reactivity to platelets or red blood cells. And that's really the area of concern that you're most worried about when you inject particles into the blood. Is it going to create clotting of some sort or interact negatively with red blood cells? And this clearly does not. Next slide. I'm now going to turn the presentation over to my colleague Bruce to talk to you about the role of neutrophils in disease, and about our lead program on the Panther platform, ASX100.
Thank you, Mark. In order to appreciate the need for the neutrophil targeting therapeutics that Mark discussed, it's important to understand a little bit more about the neutrophil biology and their role as first responders or defenders against viral and or bacterial infection, and how sometimes this can go haywire and cause morbidity and mortality. In a normal functioning neutrophil, they're circulating throughout your systemic blood circulation, and they might get signaled to go into a tissue because there might be some low-level inflammation caused by invading bacteria, or sometimes there's virus gets into the cell and the neutrophil will recognize it, secrete some enzymes to help destroy the infected cell. And what you end up having is resolution of the inflammation and tissue homeostasis, or gives the animal a chance, or the person a chance, to have its second line of immunodefenses and promote healing. And this is how it happens for normal health. However, sometimes this goes awry. When the infection is too great, when the infection is too great, the neutrophils are called to the tissue, and they're able to transgress into the interstitial or the tissue space. There they get more activated, they'll capture more bacteria or start eating cells or destroying cells that are infected, and start sending out a lot of signals, chemical signals called cytokines, which start recruiting more neutrophils and more cells to the area. Other things that are secreted by the neutrophils cause the vessels to become leaky, so the tight space between the cells opens up, which will allow fluid from the systemic circulation to get in, and you get tissue edema, and this is how you start getting edema within your lungs. This positive feedback loop, which enhances inflammation, continues recruiting macrophages and neutrophils, and causing leaky vessels, causes tissue damage. Additionally, the cytokines that get into the systemic circulation can also cause a signal to get to the liver, which you get your acute phase response, and then the cytokine storm. Anyone who's been listening to the news over the last 6 months or 8 months with COVID has heard a lot about the cytokine storm, which has been in relationship to the extreme morbidity and mortality of COVID. Some companies have tried therapeutic trials with known agents against IL-6 and I believe TNF, and the results were not good because there are a lot of other cytokines that are secreted during which confer systemic inflammation, and just getting rid of one or two of them will not allow the body to heal. So this brings us to a more broad-based approach, and which also brings us to our compound, ASX100. ASX100 is a novel microspherical particle made of polyaspirin, which has a size of about 1 micron. It is the backbone of our Panther platform. It is designed to be recognized by neutrophils, to be specifically target neutrophils. They believe it's a bacteria, and they engulf it, and this is sort of like a Trojan horse response or system where the neutrophil feels it's taking up the bacteria, but instead it's getting a bolus of salicylic acid, providing an intracellular high concentration of this anti-inflammatory, which would make this for any type of systemic inflammation, but especially for acute lung injury, ARDS, and COVID. Our hypothesis is by reducing the neutrophil response, we'll allow the animal to gain, or the person to gain control of the inflammation and begin their healing response. In the next 3 slides, we're looking at a model of acute lung injury, which is with ASX100 in the treatment mode. So the mice in the next 3 slides have been inoculated with live bacteria, which in standard state, these animals will all be dead within 48 hours. 18 hours into the experiment, the animals are then dosed with ASX 100, where the animals at this point are very sick. They're beginning to show signs of acute lung injury. But we wanted to treat this more like a patient who's going to be home for a while until they feel sick enough to go to the doctor or to the hospital. And as you can see, and this is at 6 hours after our injection, there is a 50% decrease in the amount of neutrophils that are in the lung fluid. When we reduce the neutrophils, we're also looking at the cytokines within the lung fluid. And as you can see, for the 6 top studied cytokines that are in systemic inflammation, from IL-10 to IL-6 and TNF, you have approximately a 90% decrease in the amount of cytokines present within the lung fluid 6 hours after you're dosed with ASX 100. So instead of having a very specific approach against TNF or IL-6, we have a pan approach, reducing all the markers of systemic inflammation. If our hypothesis proves correct, there should be some sort of impact upon morbidity and mortality of these animals. And as you can see at the end of the study, where after 2 days, 100% of the non-treated animals are dead. 80% of the animals not only survived, but they thrived, and they were alive all the way through the end of the study, where there was no longer any markers of inflammation or bacterial involvement. So the following cartoon summarizes what we think the state of our mechanism is at this time. With the injection or infusion of ASX 100 into a patient who is already exhibiting signs of sickness from acute lung injury, ARDS, or again, COVID right now, we are able to enter the neutrophils, phagocytize or eat the particle, which does one thing immediately. It stops the neutrophil from adhering to the vessel wall, which stops the ability for the neutrophil to actually enter the interstitial or the tissue space. When you no longer have the increased activated neutrophils getting into the tissue that was damaged, you're no longer sending out signals to recruit more neutrophils, and you're not sending out cytokine systemically, which would cause the cytokine storm. So at the end of all this, we're able to quiet the neutrophil, so it's no longer an inflammatory event, stop the recruitment of future neutrophils, decrease local and systemic inflammatory markers and responses to allow the secondary immune response to come in and heal the patient. And Mark, back to you.
Great, thanks, Bruce. Let's talk a little bit about the development plans and where we are for ASX 100. Next slide. In terms of the current timelines and milestones, we're currently in the process of manufacturing drug for clinical trials. We're going to be launching our late preclinical stage toxicology studies in the coming months, and plan to enter our phase 1 trial by the middle of next year. That study should have readouts both later next year as well as in very early 2022, which is going to give us human proof of concept data. We'll then be ready to launch into phase 2 and get that readout as well during the course of 2022. And you may ask, why is all this happening so quickly? We've purposely chosen to start with an acute indication because the times of the clinical trials and the readouts are much, much faster than if we were going after a chronic condition. And that's very helpful because it really ASX 100 is the proof of concept for the Panther platform. And so the goal here is to validate that this drug is going into neutrophils, lowering cytokine levels, doing the things that we say it's going to do, and thereby open up the broader Panther platform and additional drug programs. In terms of funding, we have a good chunk of this funding already lined up, but it's $2 million of funding to get into to finish up our IND enabling studies and get to the point of entering clinical trials. It's an additional 4 to pay for the phase 1, an additional 5 to pay for the phase 2. While that's going on, we plan to spend about $1 million on additional platform expansion opportunities that go well beyond ASX 100. Next slide. Here's just a very brief overview of what our clinical development plan for ASX 100 is going to look like. The phase 1 study, which is sort of the safety related trials, are broken into 3 parts. A part A, which is a single ascending dose in healthy volunteers, and a phase 1B in healthy volunteers as well, but a multiple ascending dose. That should help us lock down the number of times we can dose the patient. It will also help to provide proof of concept that the drug is going into neutrophils, as we've seen in animals and as we've seen in tissue samples. The phase 1 part C will actually be in a patient population where we should be able to additionally see changes in cytokine levels. So that will really provide human proof of concept. That will then set us up well for a phase 2A. Next slide. This is a very important slide. It really lays out what the competitive landscape looks like in acute respiratory distress, which is the first indication that we're going after with ASX 100. And what you'll see from this is that none of the other companies out there are quite doing what we're doing. If anything, they're taking very different approaches that could be complementary to the ASX 100 program. First off, there are the non-immune system targeting drugs. Many of those are acting directly on the lungs and trying to provide some sort of relief there. Then, as Bruce had mentioned earlier, there are many drugs targeting specific cytokines. And the problem with many of those therapies, as we have seen, is that you may need to inhibit multiple cytokines to have a therapeutic effect, and blocking any one cytokine may not work. And finally, there are other programs that are not targeting neutrophils at all, but targeting other parts of the immune system. And those could very likely be complementary to what we're doing. Additionally, there's some real validation for the indication by looking at the stocks of many of these publicly traded companies. The average stock price of a company focused on ARDS has gone up by 50% this year. And obviously, COVID has helped to shed more light on the debilitating nature of this disease and the huge unmet medical need that goes well beyond COVID to things like flu, pneumonia, chemical exposure to that could harm the lungs, etc. And we think that, frankly, our drug could be complementary to many of these other proposed therapeutic options, and that we may also see a really robust response in the public markets when we get there because of the focus on this indication. Next slide. And just to summarize ASX 100, a couple of key points. Number one, it's a very versatile drug. It'll work regardless of whether we're dealing with a viral insult, a bacterial insult, a chemical insult to the lungs. Shouldn't have any impact. We're affecting the neutrophils and, to a lesser extent, monocytes that are in the blood and preventing them from ever going to the lungs and causing a cytokine storm. Secondly, this is an acute response and a rapid response, which is what we need, because patients are either going to live or die from ARDS in a very short period of time. And if you can't get a rapid response, you're not going to help the patient. Third, it's a very nuanced modulated response. This is not like treating somebody with a steroid or, for example, dexamethasone, which shuts down the whole immune system. Rather, this is very targeted at certain immune cells that are getting into the lungs that shouldn't be there. It's still allowing the parts of the immune system that are that are in the lungs and actually clearing whatever pathogen might be there. They're still doing their job. Fourth, it's as kind of noted, it's extremely targeted. We're really focusing on the neutrophils and really downregulating the hyperinflammation that they cause. And finally, we think this is going to be a very safe program. The active ingredient here is salicylic acid, the active ingredient in aspirin. And as I kind of alluded to earlier with the overall Panther platform, there's kind of many reasons to believe this will be a safe program. Next slide. Let me talk a little bit about the investment opportunity, not just with ASX 100, but with Panther more broadly. The Panther platform is far more than just ASX 100. It allows us to design multiple particles of different shapes and sizes to target different immune cells and different immune responses. And that's really important because there are lots of inflammatory and immune diseases out there. They're caused by multiple different cell types, and they require different therapies to target for different treatment purposes. Additionally, there's a real opportunity here, and we've explored this already with particles loaded with dye, to actually take our particles from the Panther platform and load them with other therapeutic drugs besides the salicylic acid, which is a core component of the Panther platform already. And those kind of drugs, for example, could very well be steroids that could lead for a much more potent therapeutic option for certain diseases. We'd also be open to collaborating with other pharma companies and loading their drugs onto our platform for delivery to neutrophils, monocytes, or other immune cells. And the goal with ASX 100 is that it should be a very safe program. It has some really good efficacy data, and we think this will allow if we can get this one through into clinical trials and show through human proof of concept data that it works, it really opens up the whole Panther platform for much deeper development. Next slide. Let me talk briefly about the current funding situation at the company. We're currently nearly complete with our seed round. That round is at a 15% discount to the first price round, which will be the Series A. This is a safe note round. It's at an $8 million cap. This round, which is about $2 million in size, is going to get us to the start of clinical trials. We already have over $1 million committed, and much of it in the company already, from VC investors, many of whom are re-upping in a more meaningful way in the Series A. We can fit about 800K more of availability in this round. We don't necessarily need quite that much money, but we may take it to the extent that those are committed investors that'll be able to play an important role in our Series A financing. And that Series A can either be a $5 million round, which will get us through our phase 1B, so through all of our phase 1 trials, or we may raise up to $10 million to pursue through our phase 2A trial. Next slide. And that gets us back to summary of where we started, right? That really, I hope you took away a little bit about the Panther platform and its importance in delivering targeted therapeutics to neutrophils and other immune cells. I hope Bruce and I were able to convey the importance of ASX 100, which is our lead program that is rapidly advancing into clinical development and should be a good therapeutic option initially for acute respiratory distress, but frankly, we're exploring that for other acute indications as well. And then finally, we hope you've gotten a chance to appreciate the real breadth of the Panther platform, which should allow for a number of ways to monetize the platform, both through partnering specific programs on the platform, licensing the technology to other pharma companies so that they can deliver their drugs in a targeted fashion to specific immune cells. And finally, through an IPO, which we think is very realistic in light of the breadth of the platform and frankly, the high profile of the first drug and the first indication. So thank you for hearing our story, and we're happy to take questions.
Mark, thank you. Let's go ahead and jump into Q&A. Again, to all of our attendees, please use the questions chat option on your screen to send over those questions so we can answer them. Okay, let's see here. Just jumping right in. Give me one sec. Okay, so Mark, you can kind of see who this is. This better to answer either yourself or Bruce. But first question, what makes you confident that the effect you are seeing in the animal models isn't just caused by the effect of the salicylic acid on the body rather than targeting effects to the platform?
And Bruce, why don't you address this one? I know we think we have a slide that really addresses that head on.
In the same experiments, we've also utilized just aspirin at a very high dose, and it does not have the same protective qualities as ASX 100. And the other part, since we did see the reduction in neutrophils, it shows that we are very targeted with our delivery.
Great. Bruce, do you want to show the slide that has the data on that even? Do you have that handy? Should be at the... There it is.
All right, so here is ASX 100 for the number of neutrophils in the lung fluid versus aspirin. And that's basically what you want to see, is that we are seeing a reduction of the neutrophils only with ASX100.
Great, thanks so much, Bruce. Appreciate that. Next question, can you please describe the patent portfolio in some greater details?
Sure. So I think Bruce jumped to the perfect slide here. The patent portfolio incorporates both Panther as well as ASX100. The claims are around really any particles that are made using poly A in a whole wide range of sizes and shapes. And frankly, we even have some data using other shapes besides just the spherical shape, which are used for different targeting purposes. The claims also include the actually putting even targeting agents on the particles to the extent we ever want to do that to target specific tissue. That's also covered by our patents, and our patents also cover even loading the particles in different ways for the treatment of disease. So it really covers the full breadth of where we think the Panther platform will go. And frankly, to the extent as we build specific programs on the back of the Panther platform, we'll put additional IP around those particular therapeutic drugs that will obviously relate back to the patents that we have in place today.
Great, thank you, Mark. And Sue, if you had any other questions regarding that, or if we didn't answer all the way, please go ahead and chat over again. Jumping to next question for now, if we could have you explain more about the potential ways you may seek to monetize the Panther platform.
Sure, and Bruce, can you just go back to the slide on where we talk about the different ways we use the Panther platform? A couple of slides higher up. There we go. You just passed it. There we go. So there are really multiple ways that we can monetize the platform. First off, we're planning on many of our own therapeutic programs by changing the size and shape of the particles. We know that rod shapes and round shapes, for example, are used for different targeting purposes. Different size particles, smaller particles can target certain cells like a B cell. Bigger particles are needed for cells like neutrophils. Additionally, if we use very large size particles, we can actually use them to allow for depot drug delivery as well. And we're looking into that in the context of arthritis. By the way, all of that is also covered by our patents to the extent that relates back to that previous question. We're also looking at loading our particles with drugs. In the case of what we're looking to do within Asalyxa Bio, those are mostly well-established drugs that are generally the standard of care, but that may have some nasty side effects like steroids. But we are very open to partnering with others who may have new chemical entities that they want to get to certain immune cells in a targeted way, and want to do so without creating a lot of inflammatory issues or other problems that many other more traditional antibody type of targeting typically causes. So this is a much cleaner and safer way of providing that kind of drug delivery. And so we think there's multiple ways to partner these programs. There's ways for us to get many of these programs through on our own to at least later clinical trials, where we'll likely work with some large pharma company on the commercialization side. And we've had some of those early conversations. And then finally, we're looking to IPO this company within the next few years. Definitely at least waiting until we have human proof of concept data, and then it'll just be a matter of where the markets are and when we think it's the best opportunity for the company.
Thank you, Mark. Let's see here. Next question. What types of investors have participated in funding to date?
Yeah, so we've had a really strong group of investors that have come into the platform. It's really been mostly limited to venture capital firms. It's been mostly institutional money. There is one very large, very sophisticated family office that does a lot direct investing into biotech that's participating here as well. But what's great about having that institutional support is many of those investors are planning on playing a meaningful role in the Series A. So we expect we have at least several $1 million already committed, essentially already committed to the Series A from our existing investors, which should make the next rounds that much easier. And the nice thing is also is at least some of that capital, some of those funds are Midwest based. This is an Ann Arbor company that we're very committed with this company to helping to build a local Michigan biotech ecosystem. And we're very excited to be partnering with some of the local funds in building up this company.
Thank you, Mark. Okay, next question. Where do you plan on taking ASX100 after ARDS? And what other programs are you thinking about for the Panther platform?
Sure. So with ASX100, and Bruce, you may want to chime here as well after I give it some really high level color. We think this could be valuable in a wide range of diseases. It's likely a very safe program and seems to have a really broad effect on neutrophils. So any place where neutrophils are having a strong effect on the body, this could be a very good additional therapy to bring into the picture. We're starting with acute conditions, so that seems like the best place for us to start. After ARDS, we're looking at hepatic injury, we're looking at acute kidney injury, we're looking at endocarditis. There's numerous other acute diseases where we think this could have a differential impact, and if it works in ARDS as we think it will, it should have an impact in these other conditions too. From there, there's many directions we could go. I know one that the team is particularly excited about is in arthritis. There's definitely a lot of data to suggest that certain immune cells in the joints play an outsized role in the inflammation there. We also know that things like aspirin can be helpful just in reducing inflammation, but that's systemic. And we think we could maybe even create a sub-Q version of ASX100 for arthritis. So there's a lot of directions that just ASX100 alone can take. Lindsay, what was the other part of your question?
Yeah, let's see here. The second part of that question was, what other programs are you thinking about for the Panther platform?
Great. So, and I kind of alluded to this before, but we're looking at other size particles to target different immune cells, as well as different shaped particles to target specific immune cells. That's one element. And then the second wave of that is we are looking at loading our particles with other drug, steroids and other NSAIDs are sort of the first likely contenders for some of those loading programs. And that's something that we're looking to explore at the same time that we're taking ASX100 into the clinic. So we're growing the platform at the same time that we're pushing the platform forward and turning it into a clinical stage platform.
Great, thank you, Mark. That seems to be all the questions for now. Again, if there's anything else that you would like to ask the team, please chat those over to us. But for now, Mark or Bruce, is there anything that you would like to add?
Lindsay, I think we covered it all pretty well today. Very excited about the prospects for the company and the rapid potential or the rapid path of progress that we've made to date. Very excited about launching into clinical trials next year, and obviously looking for more excited and motivated investors that want to be part of the story. So eager to have those conversations.
Great, thank you so much, Mark, and thank you for joining us today. You as well, Bruce. This is Lindsay again with Family Office Networks. To all of our attendees who logged in today, we thank you very much. I will be following up with everyone individually, so if there are any additional questions or you would like direct contact with the team, I will be more than happy to do that. We hope you have a great day, and thank you so much.
Thank you.